The bacteria that we are going to study about today is Streptococcus. Now this genus called Streptococci are gram-positive cocci, and they are arranged in chains or pairs. Here in this picture you can see the small circular arrangement of the bacteria. Like this one which you see, they are present in pairs. Here you can see they are appearing in chain. So in both of the formation, you would find the Streptococci to be arranged. They are gram-positive bacteria. Okay, and the shape is circular, and that is why they are called as cocci. This organism, they are part of the normal flora of humans and animals. Some of them are human pathogens. The most important of them is Streptococcus pyogenes, which is responsible for causing pyogenic infections with a characteristic tendency to spread, as opposed to staphylococcal lesions, which are typically localized. That means this genus Streptococcus, you would find many species of Streptococci, amongst which there are a few which are pathogenic, causing diseases in human beings. One such important bacteria which is responsible for causing human diseases is known as Streptococcus pyogenes. This Streptococcus pyogenes, they are responsible for causing pyogenic infection. That means you would find them to be present in lesions, okay, pyogenic pus-forming lesions it will form. But the difference when we studied Staphylococcus, we found that Staphylococcus is also responsible for causing pus-forming lesions. But the difference in between Streptococci and Staphylococcal lesion is that Streptococcus lesion, they have a tendency to spread. Okay, but then Staphylococcal lesions, they are localized. Localized means they remain restrained or constricted to only a small space. Okay, so that is how it is. The Streptococcus is also responsible for other diseases like non-suppurative lesions. That means lesions which do not cause pus formation, then acute rheumatic fever and glomerulonephritis. Okay, this kind of symptoms you will often see as a sequel to infection. That means once an infection occurs, after the infection starts in the body, later on you would find this kind of symptoms appearing in that person. So these are all streptococcal infection. Now we see the classification. That is, what are the different types of streptococci? Streptococci itself is a genus. Now within that genus Streptococci, first of all, in order to study about the different species, you will have to classify the bacteria depending upon the requirement of oxygen. That means certain Streptococci, they require oxygen. There are a few who do not require, who can survive in absence of oxygen. The ones which are obligate anaerobes, that means they do not require oxygen at all. That is present on the right side of this flow chart. The name of such an obligate anaerobe is Peptostreptococci. Fine. Then on the other hand, you have the aerobes and facultative anaerobes. That means these are oxygen-loving bacteria, and a few who may slightly be present in absence of oxygen also for a short duration. So those kind of bacteria, they have a property of hemolysis. Remember that Streptococci who performs hemolysis may be either aerobic or facultative anaerobic. They cannot be obligate anaerobes. Hemolysis means the property to break down RBCs. So within that, you will find there are three types of bacteria, or there are three different species which comes under this. The characterization of hemolytic bacteria are done, and then they are named as alpha, beta, and gamma. The alpha, which are called the viridans group, they are classified into species based on the physiological and the biochemical properties of those bacteria. Then the next one, the beta, also called the hemolytic streptococci. They are characterized in this way as group A and group C. Okay. Here the group, actually they are divided into 20 groups, which is known as 20 Lancefield group. This is the beta streptococci. They have been divided into 20 Lancefield group, named as A, B, C, D, E, F, G, H. Then there is no IJ. Directly you find K, L, M, N, O, P, Q, R, S, T, U, V. Now, the group A, they are called the Streptococcus pyogenes, about which I had mentioned. And then the group specific C, they are carbohydrate, they possess carbohydrate antigen. Then the differences can also be based upon the serological typing, which consists of M protein. Those bacteria, they consist of this protein called M protein. These are the beta hemolytic streptococci. Then the third one is called the gamma, the enterococcus group. Enterococcus means, entero means intestinal or stomach, related to stomach. So these are related to the intestinal area. They are classified into species based upon the physiological and the biochemical properties. So let us just study in details about them. Several systems of classification have been employed, but in medical bacteriology, the following method is useful. Streptococci are first divided into obligate anaerobes and facultative anaerobes. The former are designated as peptostreptococci and are discussed in a later chapter. Okay, the aerobic and the facultative anaerobe streptococci are classified on the basis of their hemolytic properties. Fine. Then a person called Brown in the year 1990 categorized them into three varieties based on the growth in 5% horse blood agar pore plate culture. This was the person called Brown in the year 1990. He categorized the streptococci into these three varieties. How did he do this? He took horse blood and then he mixed it with agar, nutrient agar, prepared this horse blood agar 4 plates, 5% horse blood he had taken, and then he had prepared this media, special media for growing the streptococci. Then on letting this bacteria grow on the horse blood agar plate cultures, he found different kind of hemolysis. Now, what were the different kind of hemolysis that he observed? The first one, which you saw, is alpha hemolytic streptococci. The alpha hemolytic streptococci, they produce a greenish discoloration with partial hemolysis around the colonies. Okay, so that means when he had observed the hemolysis, he saw only a partial hemolysis. The hemolysis was not complete. That means that area which was discoloring was not completely white. Okay, there was a little slight discoloration and the color was greenish discoloration, which was seen around the colonies that were growing. The zone of lysis is small, just 1 to 2 mm wide, with indefinite margins, and unlysed erythrocytes can be made out microscopically within this zone. So in that small area, if you observe under the microscope, you will find the erythrocytes, a few of them which are unlysed are also present. These are known as viridans streptococci or Streptococcus viridans, such kind of bacteria. The word viridans, that means green, and that is the reason why this bacteria has been named after that color as Streptococcus viridans. The alpha streptococci are normal common cells in the throat, but may cause opportunistic infection rarely. Okay, so if you go for a trial, if we collect throat swab, normally we might find the presence of the Streptococcus viridans from our throat, as it's a normal flora, but sometimes it may also cause infection because they are considered to be opportunistic bacteria, opportunistic infection causing bacteria. Another example of alpha hemolytic streptococci is pneumococcus, named as Streptococcus pneumonia. This is also an alpha hemolytic streptococcus. The next one is called beta hemolytic streptococci. The beta hemolytic streptococci produces a sharply defined clear colorless zone of hemolysis, which is around 2 to 4 mm wide, not meter, millimeter wide, within which red cells are completely lysed. Now this one, when you see the zone around them, this is wider compared to the alpha hemolysis. And then the red cells here are completely lysed. Okay, the area that has formed the zone, that area is clear and colorless. The term hemolytic streptococci strictly applied only to beta-lytic strains. Most pathogenic streptococci, they belong to this group. You will find that the bacteria responsible for hemolysis, that is beta hemolysis, are mostly pathogenic streptococci. The third one is gamma or non-hemolytic streptococci. So here, the gamma streptococci, they do not cause any hemolysis. So you will not see any change in the media when they grow, except that the colonies you will find. And then there will be no formation of zones around the colonies. So they are sometimes referred to as indifferent streptococci. This one, they include the fecal streptococci, which are known as enterococci or Streptococcus faecalis, and related species. They are called the enterococcus group. Okay, because most of the time they are present in the intestine, and then you would find them to be released along with the fecal matters. This hemolytic streptococci were classified by Lancefield. A person called Lancefield helped in the classification of streptococci, hemolytic streptococci, in the year 1933, serologically into groups based on the nature of a carbohydrate antigen on the cell wall. So on the cell wall of this bacteria, you would find the presence of a carbohydrate antigen, and based on that, what the nature of those carbohydrate antigen are, based upon that, these groups were formed. And so this grouping system is known as the Lancefield group, where you will find 20 identified groups, and they have been named as A, B, C, D till V, but there is no I and J in between. And I had already mentioned that. The great majority of hemolytic streptococci that produce human infection, they belong to group A. Then hemolytic streptococci of group A, they are known as Streptococcus pyogenes. This may be further subdivided into types based on the protein antigen. The protein antigens are M, T, and R. Based on these antigens which are present on the cell surface. This is again according to Griffith typing. Griffith was another scientist, so according to his calculation, this subdivision was made of M, T, and R antigen that was found to be present on the cell surface. So about 8 types of Streptococcus pyogenes have been recognized as far, and the types are 1, 2, 3, and so on. Okay, so this was in general about Streptococcus. Now, when we need to study about particular Streptococcus, we are going to study about Streptococcus pyogenes. Streptococcus pyogene, as I told you, is responsible for causing pyogenic infection or pyogenic lesion. This particular cocci The size is 0.5 to 1 millimeter in diameter, whereas they are spherical and oval in shape. They are arranged in chains because chain formation is due to cocci dividing in one plane and daughter cell failing to separate completely. That is the reason why the cocci, they all come under one chain. Otherwise, you'd find them in pairs. Streptococcus has been classified as Streptococcus longus because of the long chain, and some are known as Streptococcus brace because of the short chain. Streptococcus are gram-positive, non-motile, non-sporing, and capsulated. The culture characteristics, it is aerobic and facultative anaerobic, and they grow best at 37 degrees Celsius. It is exacting in nutritive requirements. You will find them to be occurring or growing properly in media which contains fermentable carbohydrate that ferments. Okay, and then they are the media can be enriched with blood and serum, and then you see them growing very well. When cultured on blood agar, it gives colonies which are circular, semi-transparent, low convex disk with area of clear hemolysis around it. So that is, you will find beta hemolysis occurring when this bacteria grows on blood agar. Virulent strain on fresh virulent strain on fresh isolation from lesion produce a mat colony, while avirulent strain from glossy colonies. So this is a characteristic identification. If you, based on the colonies that you see on the culture plate, if you want to identify the bacteria, whether it's a virulent strain or avirulent strain, how are you going to see that? How are you going to identify that? If you are isolating a bacteria from the lesion and then growing it on the culture media, and when the colonies form, if you see the colonies are finely granular and matte. Matte means they're not shiny, okay? Those are known as matte. So that those will be virulent strain. That means those strains are still dangerous. They still have those power to cause disease. Then while avirulent strain, they are shiny, glossy colonies. Okay? Strains with well-marked capsule produce mucoid colonies. There are some strains which may have capsule around them, and those colonies are mucoid colonies. When the colonies are mucus-like, so then you can understand that these strains have capsule around them. When cultured in liquid medium like glucose serum broth, it shows granular turbidity with powdery deposit. Okay, and no pellicle is formed. These are the medically important streptococci and their characteristics. You can go through it when I share this note with you. Then the biochemical reaction of this bacteria, it ferments all sugars like sorbitol, maltose, lactose, and mannitol, producing acid but no gas. It is catalase negative. This particular bacteria is resistant. It's a very delicate organism. When if a heat of a temperature 54 degrees Celsius is provided for 30 minutes, then it is easily destroyed. It dies in a few days in cultures unless stored at a low temperature, that is 4 degrees Celsius. Preferably if you grow them in Robertson's cooked meat media. Then it can, however, survive in dust for several weeks if they are protected from sunlight. It is rapidly inactivated by antiseptics. It is more resistant to crystal violet than many bacteria, including Staphylococcus aureus. So this is the property of resistance of this bacteria. Okay, so what are the things they're resistant to? What things can kill them has been mentioned here. Then toxins, enzymes, and other virulence factors. Streptococcus pyogenes produce several types of exotoxins and enzymes. And this exotoxin and enzymes, they act as the virulence factor. Also, young protein act as virulence factor by inhibiting phagocytosis. Young protein means when when the bacteria is young and the protein that has been developed in that is not matured yet. It is in a very nascent stage. So that kind of protein has got the virulence factor, and then it can inhibit phagocytosis. The C polysaccharide has been shown to have a toxic effect on connective tissue in experimental animals. So they have got this two properties. Streptococcus, they produce two hemolysin. Hemolysin, which helps in the lysis of the RBCs. The two types of hemolysin are streptolysin O and streptolysin O and S. Okay, so these are the two types of hemolysin that are produced by streptococci. The streptolysin O, it is called the alphabet O has been used to name it because it is oxygen labile. Okay, it is inactive in the oxidized form, but may be reactivated if they are treated with mild reducing agent. On blood agar, the streptolysin O activity is seen only in the pore plate method and not in the surface culture. It may be obtained in the active state by growing the streptococci in broth containing reducing agents such as sodium hydrosulfide. It is also heat labile. It appears to be important in contributing to virulence. So if any virulent factor is considered, streptolysin O can be considered as a virulent factor. It is lethal on intravenous injection into animals and has specific cardiotoxic activity. That means this can affect the heart. It has leukotoxic activity also. The leukocytes will also be affected. And in its biological action, streptolysin O, it resembles the oxylabile hemolysin, which is present in Clostridium perfringens, Clostridium tetani, and pneumococcus. So it is quite similar to the streptolysin O that is secreted by perfringens, tetani, and pneumococcus also. Then this streptolysin O is antigenic, and anti-streptolysin O appears in serum after streptococcal infection. So when a person is infected with streptococcus, if we want to confirm the presence of streptococcus in that person, what can we do? We can perform a test which determines the presence of anti-streptolysin O. Okay. Because after a person is infected with streptococcus, if the streptococcal infection starts, then this anti-streptolysin O will appear in the serum of the person. And this is an important diagnostic procedure for streptolysin O, or you can say streptococcal infection. Then streptolysin S is so called because it is soluble in serum. This shows stability with oxygen, dry heat. It is responsible for hemolysis seen on the surface of the blood agar plates. It also has leukocidal activity. Then another toxin which is called pyrogenic toxin, pyrogenic exotoxin. It is a toxin which is named as erythrogenic because it is intradermal inoculation in susceptible individual causes erythematous reaction. That means if you want to again check a person whether is suffering from this streptococcus, and then if there is the presence of this pyrogenic exotoxin in that person, then what can we do? We can do a test. And that test is inoculation, intradermal inoculation. Intradermal is within the skin. Intradermal inoculation of this toxin into the person, into the individual. If it shows erythematous reaction, that means if it shows some skin reaction, that means it is it will mean the presence of this exotoxin in that person. This test is known as Dick test. This test is used to identify the children who are susceptible to scarlet fever. Okay, because, and this name scarlet fever is because of a toxin which is called scarlatinol toxin. This toxin, it induces fever, which is named as streptococcal pyrogenic exotoxin. It has three types, A, B, and C. It is a super antigen, so it can act as T cell mitogen and causes rapid release of inflammatory cells, which cause widespread manifestation. There's an enzyme which is called streptokinase enzyme. It acts as a toxin again, and it promotes the lysis of human fibrin clot. That means when a person bleeds, what happens? After some time, there is the blood that the place from where the person starts bleeding, that area, from that, and at that area the blood gradually clots, right? So this is because of this production of fibrin, which happens to help in the clotting of the blood. But then this enzyme called streptokinase, which may be released by this Streptococcus, it is a toxin. It acts as a toxin, and it promotes the lysis of that clot, that human fibrin clot, because it activates plasminogen to plasmid. And then when this happens, this acts as a diagnostic marker, as it antigenic in nature. So antibodies are produced in convalescent area. This is an antigen, right? So then because of the presence of this, you would find the presence of production of antibodies in the body also. This is helpful in the retrospective study. So these are some of the toxins in the form of enzymes that are released by this bacteria. Okay. The antigenic structure. Antigenic structure, besides toxin, the bacteria also has some other antigenic structure which helps them to cause disease or infection in a host. First of all is the capsule. It is a capsulated bacteria, so it has a capsule around that, and that capsule, when it is present, it inhibits phagocytosis. It is not antigenic in human beings. The cell wall is composed of an outer layer of protein and lipoteichoic acid. A middle layer is present, which has a group-specific carbohydrate, and an inner layer of peptidoglycan. The peptidoglycan is responsible for the rigidity of the cell. It also has some biological properties such as pyrogenic and thrombolytic activity. Serological grouping of streptococci depends on the C carbohydrate, which I told you. Then Streptococcus pyogenes, they belong to which group? A, the group A. And this antigen is an integral part of the cell wall, it has to be extracted for grouping by a precipitation test, with group antisera. Okay, then this Streptococcus pyogenes, they are again differentiated based on some protein which is called surface protein. The surface protein are protein M, T, and R, which are present on the surface of the cell. The M protein is the most important of this all. The M protein acts as a virulence factor by inhibiting the phagocytosis. It is antigenic in nature. The antibody to the M protein promotes phagocytosis of the coccus and is therefore protective. The antibody which is produced for this is protective. Okay. Then it can be extracted by the Lancefield acid extraction method, and the typing is done with type-specific sera. The next type of protein, the T protein. It is an acid labile protein, and it is resistant to trypsin. It may be specific, but many different M types possess the same T antigen. Then it is usually demonstrated by the slide agglutination test using trypsin-treated whole streptococci. You would also find the presence of hair-like pili or fimbria, which are projecting through the capsule of group A streptococci. The pili consist partly of M protein and are covered with lipoteichoic acid, which is important in the attachment of streptococci to the epithelial cell. So that was about the antigenicity. Now, when we talk about pathogenicity means what type of diseases are caused. So this Streptococcus pyogenes, they produce mainly two types of lesion. One can be suppurative, another can be non-suppurative. The suppurative infection may be caused in respiratory tract, skin and soft tissue, then genitalia, abscesses in liver and lung, kidney and brain. And the non-suppurative, they are acute rheumatic fever, acute glomerulonephritis. In the respiratory tract, this is the primary site of invasion of the human body by Streptococcus pyogenes, and the area is throat. Okay, the sore throat is the most common of the streptococcal disease. It may be localized as tonsillitis, or it also can involve the pharynx if it's spread. Okay, from tonsils, if it starts spreading, it can affect the pharynx, and then it can cause pharyngitis after that. The virulent group A streptococci adhere to the pharyngeal epithelium by means of lipoteichoic acid ligands. Then tonsillitis is more common in older children and adults than in the younger children, who commonly diffuse pharyngitis. Then localization is believed to be favored by hypersensitivity due to prior contact. Next is chronic tonsillitis. From the throat, the streptococcus, it will spread to the surrounding tissues, then it will lead to suppurative complications, which will affect the otitis media, which will cause otitis media. Then these are the other disease problems that will happen during chronic tonsillitis. Then next part is the skin and soft tissue infection. The skin is affected, and if a person is wounded or if it's a burn injury, then there is a high chance of this bacteria causing infection. Then it can again spread and cause lymphadenitis, causing problem in the lymph nodes, and cellulitis in the cells. Infection of minor abrasion may at times lead to factor septicemia. That means, you just might have just a skin bruise, okay, and in that area, if that bacteria has caused an infection, if not treated properly, the infection from this minor one may become fatal. Then there's another streptococcal infection in the skin, which is known as erysipelas and impetigo. And erysipelas, it is the another type of problem, which is a diffuse infection. Diffuse means it spreads, okay, involving the lymphatics. The affected skin, which is red, swollen, and indurated, is sharply demarcated from the surrounding healthy area. So the area which is affected, that will become red and swollen, and that can very easily differentiate between the healthy area. While erysipelas is rare and seen only in older patients, impetigo is found mainly in the young children. The skin area shows erythema with edema. One attack does not give protection, and recurrent infection in the same area occurs in some person. Then impetigo, it is again caused by the Streptococcus pyogenes. It is a superficial crushed spot which happens on the upper part of the skin. Okay, that's why it's called superficial. And especially in children, you will find it, and the diameter of that on the skin is less than 1 inch. Impetigo and streptococcal infection of scabies lesion are the main causes of acute glomerulonephritis in the children. It also causes genital infection. It may be both aerobic and anaerobic streptococci, which are normal inhabitants of female genital tract. Then it may cause infection when they become opportunistic. There are other suppurative infection that may happen in internal organs like brain, lungs, liver, and kidneys, and also septicemia and pyemia. Then these are the non-suppurative complications like rheumatic fever and acute glomerulonephritis. Glomerulonephritis is when it affects the nephrons and the kidneys. This is another table which will give you the differences of the diseases. Okay, and the lab diagnosis, we can take the samples and then we can go for the gram staining. Then we can go do culturing of those bacteria, and then also we can determine the presence of the anti-streptolysin O, anti-streptolysin O will appear in the serum. For that, we'll have to do the ASO test, which determines the anti-streptolysin O. Then the other antigens can also be determined by using certain kits, which will help you to identify the disease. Okay, so this is about Streptococcus pyogenes.